All right, g'day, good afternoon. Thank you all for coming. It's great to see so many faces that I haven't met before. And I guess on that note, I'd sort of like to take the opportunity of this talk to introduce not only myself, Bort Edwards, but the project that I'll be undertaking here as a Smithsonian postdoctoral fellow under the guidance of Dr. Vicky Funk in the herbarium and funded by the USGS Powell Center for Interdisciplinary Synthesis. The project is still in its very early stages, so I thought I'd present some background on the methods and the expectations, not only as an introduction to this work, but as an insight into how big data is becoming increasingly available for leverage into large-scale projects and how these projects come together. So, ever since people started to take an interest in the world outside their own little local patch, they've been fascinated by the diversity and distribution of organisms and the question of why things live where they do. Why are plants and animals not distributed evenly and consistently across the landscape? So on a grand scale, there's a fairly obvious limitation that most species are just not able to travel the distances to be everywhere at once. This leads to isolation of populations and, over time, the evolution of distinct species and the formation of different communities. However, even at a local scale, when dispersal shouldn't be an issue, we observe species diversity and heterogeneity. So the question remains, why do species live where they do? Linked to this question is why do species live, well, linked to the question of why do species live where they do, is why are some groups of species so much more diverse than others? An alternative way to look at this question is to flip it upside down and ask, why do species not live where they don't? Does something in the landscape make it hostile to some species? If so, could adaptation to tolerate these conditions allow those other groups that do thrive there to expand and become hyperdiverse? As mentioned, these questions are not new. Intuitively and observationally, we expect that the drivers for plant niches will be predominantly environmental, specifically the soil that they grow in and the environmental conditions, the weather conditions that they're exposed to. And we do have confirmation from a variety of small-scale studies, usually using a small number of species, people's pet groups, that this is probably certainly true. However, this project aims to extend these hypotheses out from local studies and to use big data to identify these patterns across a larger scale. So the innovation of large collaborations is shared online databases and the availability of previously unwieldy data sets that allow us to address these questions on a scale that hasn't previously been possible. For our project, fundamental to the questions of the role of the environment in plant diversification is the first data stream that we have, which is environmental. This brings together decades of soil, geochemical and climate surveys from both the USGS and the USDA, as well as collaborators in Mexico and hopefully Canada. And one of the main challenges of our project is to integrate from these various disparate sources, the surveys and the databases, into one single data set that covers the entirety of the North American continent. So this environmental layer data will provide the basis of our study. The second data stream are records of plant distributions. Here there's an already pre-existing network of online databases. This brings together specimen and observational records from museums, universities and governmental organisations across the world. Mapping where the species have been found to occur in the environment allows us to extract information on what conditions these species can then tolerate. The third data stream is also from a pre-existing database. This is Genbank, which is an online repository of DNA sequence data that's been contributed by hundreds, thousands of researchers across hundreds and thousands of studies. And by searching the database for our species of interest, we can use these sequences to construct phylogenies, which estimate which species are most closely related to each other, and thus whether they arose and when they arose from a common ancestor. So by integrating environmental data, such as maybe sodium levels, we can then map back from where this occurs in the present day, back through to whether this may have been a feature common over the ancestors. And by inference, is it likely that the evolution of this tolerance in the ancestor has allowed for current day species to live where they do, and whether this may have driven diversification? Similar methods have been used in a pilot study in Australia, and we found that salt and alkalinity tolerance have allowed for acacias to colonise harsh habitats previously inaccessible to many species. But the drive of our project is to address North America and what habitats and environment factors appear to have played a similar role in highly diverse plant groups on this continent. In particular, the southeast of the United States and north of Mexico provides a mosaic of challenging and hostile environments, characterised by water availability, high salt levels and high temperatures, and is well known for high levels of species diversity and endemism. Yep. Okay, the next slide. As a test case, we're first going to be focusing on the compositae. These are daisies or sunflowers, with reasons for their diversity is still relatively unresolved. 1 in 10 flowering plants is a daisy. Many of you probably ate them today, lettuce or sunflower oil. And their centre of diversity is also in southwest US and northern Mexico. So once we've established methods, we intend to extend that to groups of other large plants using the collaborators from various institutions around the US. If you'd like to follow the progress of the project, the PAL Centre at usgs.gov, and me personally on Twitter or Instagram. Thank you very much. I'm Bort Edwards. I'm afraid I'm going to completely ruin the setup. I was preordained to be a biologist. My father is an entomologist, and so we spent the majority of our spare time being hauled around the landscape, looking at the geology, at the flora, at the fauna. It was kind of inevitable that in some way I would see that as not only an amazing experience and something that I could maybe do as a career, but something that I could do as a career with a parent who had managed to forge a successful one doing that. But at the same time, I still find it hard to understand why not everybody on Earth wants to be a biologist. It's so hard not to go out into the natural world and just be fascinated by what you see. I guess the story is then, how do you get to your particular niche in the scientific community? So my father, as a taxonomist, showed me that it was interesting to put things into boxes, but then I became interested in how the boxes related to each other and how they got to those boxes in the first place. Plants, because they're not insects, and bringing in other sources of data to answer the question of why we see this amazing assemblage of life around us that we do. So growing up in Australia, it's hard, again, not to be outside experiencing these things. I did my undergraduate there, I did my PhD there. I then got lost in the University of Rochester studying Drosophila genetics for 2 years until my current supervisors dragged me out of there and have sort of tried to rehabilitate me back into plants. To come from that situation where I considered giving up science back to such an amazing institution where everybody is incredibly enthusiastic and grateful to be here, where everybody's supportive, where everybody is doing what they love, is just fantastic. To the interns, just use every possible opportunity you have. Don't hesitate to come back when you've finished. Use this as an opportunity to make contacts and to find out what you want to do. Again, I don't understand why you'd want to do anything else. And if you do decide to do this, please use us as a resource.
Maybe I'm not seeing it, but this sort of horse cart problem of whether the diversification has led to tolerance or whether the tolerance has led to diversification.
Yeah, so that is an excellent question. There is a degree of horse and cart to it. There are methods that we can use to try and get around that. I guess one of the other things that we're trying to bring to that is sort of replication. So we have a large group of species and we're hoping to see the same pattern emerging in multiple different places. We're still in the process of pulling our methods together. It is a problem, but it's certainly something that there are ways out there to address, yes. Sorry, that's a very non-specific answer to that question. But yeah, if I could talk to you afterwards about that, that would be really good.
Talk was a nice illustration of the power and potential of big data approaches to I wonder about some of the challenges, in particular data quality. That worked a
My intern was just grimacing behind you. So yes, data quality is, I wouldn't say it's horrible, but there are parts of that are horrible. There is a point where you have to accept that the data's not going to be perfect, but we're trying to get it as close to perfect as we can. I've spent the last 3 weeks, it turns out that GBIF has decided to auto-synonymize species names in the database, and it's managed to lump together a whole number of species that have the same genus initial, the same species name, and the same initial of the original author. So it's lumping species across whole different parts of the family. I only discovered this because there was a species that had distribution in Alaska and in Brazil, and that struck me as slightly odd, and I realized it was calling 2 different species the same thing. So I'm now faced with the problem of going through and looking at every single species and its synonyms to make sure that it hasn't done this in other cases, and it turns out it has. That's the sort of thing, the problem with that is that's the sort of thing that doesn't get picked up by algorithms. So we do have a suite of tools that will run through and automatically flag outliers geographically, outliers taxonomically. And to some extent, we can clean the data to that extent. But there are going to be problems in the data, just trying to get it to the best that we can get it. Yeah, it's not ideal. Again, I guess we're hoping that the weight of data might the signal will outweigh any underlying dirt in the data. But yeah, it's a problem, and I think that's something that people are working on. But as big data becomes more and more used, it's something that is going to be needed more and more, really efficient, effective tools to address that. So appeal to the data scientists out there. We do need your help.